Hello, Instagram. Today we're going to be talking to Dr. Knock, who is a phenomenal science communication personality. I think he's sort of risen to fame on TikTok, but he's on Twitter and Instagram and all sorts of things. He is a PhD educated scientist who works at a big pharma, but a pharmacological company working on a cure for COVID. And this is going to be a little bit different because I think we're going to have a slightly shorter talk, and I'm going to really focus on trying to look at some of the basics of the COVID-19 virus and the basics of the biology. We're going to go over a little bit of his work on the vaccine, and probably at the end we'll get into some mushroom questions. So I'm going to get him on the horn here, and I think this is going to be a really good one. Super excited to talk to the doctor.
And I believe he'll be wearing his trademark blue shirt. So we'll see what it looks like. But hello, doctor. How you doing?
What's up, Gordon? I'm doing great. How are you?
I'm excellent. I'm glad we could do this.
Figured out here. Yeah.
Yeah,
This
totally.
is fun.
It's quite honestly, this is a little weird for me because I'm so used to seeing this view of you educating me on science in your living room, but here you are in the flesh. So
That's right. I just never leave this 3 by 3 foot, area here.
So actually, I'm curious for you because you mentioned that you devote about half an hour a day to making social media, which is incredible because you come up with these fantastic videos and you concisely communicate science in such an incredible way. And you only do this in half an hour a day.
Half an hour to an hour on average.
People might assume that you spend your entire day doing this, but no, you got a day job where you work on a vaccine for this pandemic that we're all dealing with. So anyhow, thank you for taking the time. I guess I'm curious. I also have a PhD, but I probably worked on pretty different things. I worked on like yeast biology as it related to fermentation of wine and kind of microbial interactions and community ecology of wine fermentations.
Okay, great.
So I have a good degree in background in biochemistry, but what the heck did you do, and how did you kind of shift into what you're doing now?
Sure. So the closest thing we did, at least to yeast, is we use a lot of yeast display. And so I don't know if you're familiar with yeast display as a method of designing antibodies against new targets.
Mm-hmm.
And so I didn't do this too much in my PhD, but other people in our lab would take yeast and then use them to mutate and form a bunch of different antigens, and then you choose the best immune response against it, and you can select that way. That's not what I did for most of the part. I was studying immune responses to pharmaceutics.
Mm.
And so that's a type of immune response you don't want to have happening.
Yes,
Because
yeah.
patients can have antibodies against these drugs, and then if they take them again in the future, they either get cleared very quickly from circulation, which means they can't do whatever the drug was supposed to do, or they're at higher risk of going into anaphylaxis or something.
Yeah, some sort of autoimmune shock kind of thing. Yeah.
Exactly. Yeah, very much like shock. And so we were studying ways to prevent it from happening in the first place in people who don't yet have those reactions, as well as for people who already have those reactions. What can we do to basically downplay that immune response and still be able to treat them with those drugs? And so it was very much focused on how to prevent immune responses against particular targets. And now I've kind of done the flip side now, working in pharma, where we're trying to build monoclonal antibody-based drugs against COVID right now in particular, but also other respiratory pathogens like influenza, respiratory syncytial virus, metapneumovirus. And so in that sense, it's been very easy to pivot from what we usually do, which is making drugs for respiratory viruses, and just apply that same platform to COVID, basically.
Yeah. Yeah, and all hands on deck. Let's deal with what's in front of us. So was your PhD immunology then, or?
It was technically in pharmaceutical sciences, was
okay.
the broad name for it, but it was, in studying the immunology of pharmaceutical sciences. Within pharma, there's kind of two broad halves. There's the chemists, and then there's the biologists.
Mm.
And so I'm very much in the biology side of things.
Okay.
If we need to do any chemistry for any purpose, we work with chemists. We don't try to do it ourselves.
Yeah, that's a good way to go. But I mean, science is very much, I feel like the history of science was that you had people siloed in different departments, and the geneticists and the biochemists and everybody was so sort of separated. And, in the last 20, 30 years, everything has moved to this integrated omics approach where now like everything has to be done between different departments. And, even my grad program was like a biochemistry group and a cell biology group that mishmashed together, and we called ourselves the alphabet soup group because we were B and C D B, as an impossible thing to remember.
Yep.
But so, I mean, I guess in grad school, people have very varied experiences, and PhDs are hard. There's a lot of time that goes into it. It's a slog to get through it. And I guess I'm curious, sort of, do you have reflections on your grad experience, and then like kind of what about that helped land you where you are now in industry?
Yeah, it was kind of a transformative process. As I'm sure it was for you and all people who go through grad school. Is at first, you're kind of, maybe you've done some undergraduate research, and maybe in that circumstance, people were telling you sort of, this is the project, this is what you should do to advance that project, and it's generally sort of lower risk, lower responsibility in that sense. Then you get to grad school, and it's like, okay, here's 3 kind of new projects. 2 of them have grant funding. 1 of them we're hoping to get grant funding. Figure out basically what can work. And so that's kind of daunting at first as a PhD student, but then as you get to 3rd year and 4th year, you start to own your project, and then you end up knowing more about it than your PI does in your lab, or pretty much anyone else does.
Pretty much anyone else alive, because you are the only person who cares so much about that tiny little focus. So
Exactly. And so that's another thing, like when people talk about the PhD thesis defense, I don't know how yours went, but it's not really as scary as people make it out to be, because by the time you get to that stage, you know your stuff. And so it was cool getting to know sort of what your limits are as a scientist. Was sort of the process for me. Is you start out not really knowing anything about how science actually works, but by the end of it, you do have a really good sense, at least of how academic work goes, which turns out is quite different from how industry work goes.
Very different.
And so that's been a cool process as well.
That's awesome. And I take it you're probably part of a larger group where you get to work with teams. And like you said, when you have chemistry you need to do, you reach out to the teams. And I think that's, in academia, there's a lot of collaboration and cross-department stuff, but there's not a clear path to it. So I feel like everything you do in academia, you kind of have to forge your own relationships. Whereas at a company, there is something like, oh yeah, we got a stats guy. Just feed the stats guy the stuff and you'll get back an answer, kind of thing. And so there's a real difference formalized in the timeline of way things happen. Because academia, if someone's like, yeah, I mean, honestly, I just published a paper from my last chapter of my dissertation, and it took 4 years. And partially that's because we had a lot of, had to go back and forth a bunch of times. But embarrassingly, it took me that long. And that's not atypical in academia at all, to say that took 4 or 5 years to get this publication out. But if you said, normal. Hey, we spent $80 million on this drug, and
we've got a 2-year delay, and the patent timer is ticking.
Yeah, and they're like, nope, you're done. And that's what happens in industry, is things get shut down if they're not viable, I guess. And industry, I mean, I guess one of the things I want to talk with you about is just that big pharma gets a bad rap. Right. The idea of big pharma, and there's these shadow corporations, and to some degree it's validated in the cost of drug prices and the way that things are brought to market. But at the same time, I worked sales and I went to a lot of these big pharma companies, and I got to meet guys like you. And for the most part, scientists who work there are really nice people, and they really care. That's the reason they go work for these companies, because they want to help people. And I guess, can you speak to that? You work at a big pharma company, and like, you guys are probably demonized, at least from this theoretical point of view, but like you're just people, right?
Oh, totally. And there's sort of 2 layers, and you alluded to both of those layers. There's the first layer, which is that prices are too high for drugs or something, and that very well may be the case. That's a business decision, for investors and everything. The second layer, which is a little bit more extreme, is when people start to say big pharma has the cure for cancer, or they have, they manufactured COVID so they could sell vaccines or something. And so that level is where it gets into this, idea that big pharma is just made out of individual people like me.
Right.
And you can't keep some like world-changing secret among thousands and thousands of people who are just normal people who personally don't benefit financially from unleashing COVID or something.
And even when your company does well, it's not like you guys all get some huge bonus or something like that. Everyone at a pharma company just makes their salary. That's
right.
It.
Yeah, I mean, for smaller biotechs, like maybe you do have some sort of a small equity position, but regardless, it's not like the other important point though is that pharma, it doesn't hold all of cancer research. There's research going on in universities, publicly funded, at the NIH, government funded. And so, if any single person had some big cure or something, they would become instantly super famous.
Yes.
And whereas they gain nothing from hiding it. So those types of criticisms, they don't make too much sense. Now, charging too much money for drug prices, that could be a totally fair question.
Right.
Obviously, healthcare expenses in the United States in particular are way out of control, partially driven by drug expenses, but not totally driven by drug expenses. And so that's a whole different sort of like economic question.
Right. And that gets at like the people who run your company rather than like the scientists who work there. And I think what ends up happening is the decisions of the business people at the top of the food chain cycle down and somehow get metastasized into animosity towards scientists, which is, I think, clearly a real problem in our country right now, that people don't trust scientists and somehow think that scientists are actively potentially working to hurt them or not tell them the truth or collude something. And that just, like, it scares the crap out of me. You know, like, when do we stop trusting doctors and scientists and people who pay attention to the climate? When do we stop looking at experts and say, oh, you're not an expert. My opinion that I saw on Facebook is somehow more valid than your 20 years of experience in the field.
Right. And I think that's been a couple of decades coming, that, you know, the death of expertise. People have written a lot about this, where, you know, with increasing access to information, people become increasingly confident that they are capable of interpreting that information. But having access to it and being able to critically interpret it are two totally separate skill sets, which very few people have. But it doesn't matter if they have it or not. If they think they have it, that's the deal.
Yeah, and that's like the Dunning-Kruger effect kind of come to life. And I think for me, looking back at my grad experience and really looking at a lot of school, did I learn that much specific content? Somewhat, but I'm not the best memorization person. And I'm very thankful that I live in the age of the internet, because I can have kind of a brain like Swiss cheese, but I can remember general concepts and I can go look up specifics as I need to.
That's it. You know how to look it up.
And you have the ability and capacity for critical thought. And I think one of the biggest issues, and my PI actually years ago gave a keynote speech on this, but it was about the idea of, in the information age, what we lack is curation. And in some sense, I'm very thankful for accounts like yours, because you are bringing that level of curation, but you're also doing it in this really like fantastic, exciting, you know, short format. You're doing trends and viral videos and things like that. And you make entertaining content that's conveying science in a really relatable way. And I like, thank you for that.
I appreciate that. It's definitely not the norm, though. I mean, if you think of traditional curation, it's like academic journals, right? They're separating the good research from the bad research. But then that's totally different from the ones that's published. Even if it's a reputable study in a journal, there's no limit to who writes about it on the internet. After that state, there's no regulatory impact there at all. And it's you know, whether there should or should not be some sort of like a, you know, governing curation of scientific information in media is an interesting question. We're seeing right now some of the very real negative effects of there not being any limit on people posting very, very incorrect things, claiming to be from reputable sources that are not. It kills people.
Yeah,
if you take
literally,
that away,
yeah, more than ever in history, is it killing people? It's not just like, oh, one person died because they took the wrong drug. Like, no, we have, I don't even know how many, 215,000 dead in this country because of misinformation and lack of credibility and listening to scientists. And
yeah, it's not just like, oh, those people think the earth is flat. They're wrong. We should tell them they're wrong. It's like hundreds of thousands of people are dying as a direct result of it. So it's kind of higher stakes than, you know, like the curation, the balance of should people just be able to write whatever they want and publicize whatever they want versus should it have to have some element of accuracy?
Yeah.
I mean, and that question now.
That gets at the idea, you know, free speech is free speech, yes, but free speech doesn't mean the freedom from an accountability for what you say. And I think that's what we're seeing at like a national level now, is that we need to bring back that accountability. It's okay to say whatever you want, but if you say something really outrageous, people need to be able to call you on it and hold you accountable for the fact that you lied, rather than letting it sort of move on to the next news cycle kind of thing. But while I've got you, I want to do some kind of COVID trivia. And
Let's
this
do
is
it.
a little different than what I
Let's
normally
see.
do with the mushroom stuff. But I've been tracking this, and honestly, compared to like what I was doing in like March and April, I am not paying as much attention as I was, just because mental health stuff, you can't constantly look at the ticker every day and go, oh my God, how many people are dying and stuff. But
yeah, there's no need to really.
Yeah. It's happening. This is the reality for at least the next 6 to 12 months before we get some kind of vaccine. And that's one of the things I want to talk about, is a timeline for that. So let's get into COVID. It's a virus. So that's, you know, viruses are different from bacteria or yeast or other microorganisms. In theory, viruses aren't alive from a biological definition. Where do you fall on that?
Hotly debated.
Hotly debated, right? Yeah.
I fall under not alive, but
Okay.
you can make a reasonable argument that they kind of act like they are.
They're kind of like, they live as parasites inside of other things, but without other living things, they wouldn't exist, basically.
Exactly. Right. They require that continuous chain of other living stuff in order to keep persisting.
Yeah. So the the origin of this particular virus has been very well documented. And despite lots of outrageous claims to, other ideas, it was basically found in a bat in China and probably came as a zoonotic transmission from people living in too close proximity to bats, and or eating bats incorrectly prepared or something to that effect. But there was a research institute in Wuhan with a bunch of American scientists there doing work on these viruses. And it's not like it was designed in a lab. It was just like they were sampling the local biodiversity and they'd seen this virus, basically, as far as I understand it, right?
Right. That's sort of our default understanding. And so like theoretically, it is possible to engineer a virus in a lab. There's no doubt about that. You can do gain of function studies to make them more dangerous, loss of function to make them less dangerous. But the thing is, unless you have very good evidence that that happened, given that our other most likely outcome is that it's very similar to this virus that we have documented from the wild from bats, they have gone through an intermediate reservoir, went to humans, that has to be your default conclusion as to what happened, unless you have very strong evidence otherwise.
And I think it's a good point that, we can look at the sequence of the RNA or the nucleotides and make inferences about the way that that virus has evolved. And I think at one point you posted a great story about the nextstrain.org, which is a website where you can look at the evolution of different strains of COVID, because there have been multiple different strains. And I guess in some sense, we're very lucky that this RNA virus does not mutate very quickly. Its rate of mutation is fairly low, because as far as I remember, I think it has a fairly large genome for a virus.
It does. And so its rate of mutations is actually lower than it would be for smaller ones with less of that checking machinery. And so it doesn't change too fast. That's part of the reason why we're not too worried that, you make a vaccine against this early strain, and then it's going to mutate so much by the time the vaccine comes out a year later that it's a totally different virus and the vaccine doesn't work. That's not really a concern because of exactly what you're saying. The rate at which it's mutating is pretty slow. It certainly is mutating, but not so much that the features of it that your immune system recognizes are going to be noticeably different. Right.
And that's a great point. Is, we can worry about mutation, and maybe eventually within a couple of years, there could be a slightly different strain that would need a different type of vaccine. But there's a good chance that this coronavirus may hang around, and like the flu vaccine, we may need to get a yearly dose or re-up on the antigens to deal with whatever the sort of most recent thing is.
Right. And that's a function of declining immunity rather than changing virus. Like the flu, you need an annual one for both reasons, because your immunity declines and because the virus changes. Here, your immunity is just declining.
Yeah, that's good. Good to know. It's not something I totally thought about. So, in terms of the infection mechanism, as far as I understand it, these are basically it's a bunch of RNA in a lipid capsule, which is important too, that it's covered in lipids, because that makes it survive longer than it would if it was like naked RNA. Naked RNA viruses or things that have different types of coat proteins don't survive very long outside of a body. And that lipid capsule helps keep it sort of hydrated and potentially more viable in aerosol form for a longer period of time, so that you could walk into a room where someone had been COVID positive, like, I don't know, a couple hours before. Yep. And still, if there's aerosols in the air, you could get infected. And the mode of transmission seems to be primarily through aerosols that are going through, your nasal passages or through the eye to potentially, so your eyeglasses are, you're on
slightly helpful here. Yep.
You got your built-in PPE. As opposed to it being, I think when COVID started, I watched everyone like spraying their groceries with bleach, and I was like, it seems like a waste to me. I've never,
well, it can still
wash
happen.
your hands, but it can still happen.
Yeah, it's easier to protect by washing your hands, for example, after you touch something that's infected, like a doorknob. As long as you then don't then go touch a mucosal surface, it's not going to jump from your hands to your face.
Right.
So maybe it is helpful to spray things. It's not going to be the dominant form of transmission, though. It's definitely going to be droplet cannonballs going from someone else's mouth to your eyes or to your mucosa, or aerosols that eventually build up in an area, particularly not well ventilated areas like indoors without the windows open or doors open. Mm-hmm. And you can think of it almost like a dose. One of the mistakes people make thinking about coronavirus is in like binary infected versus not infected.
Yeah, totally.
And the way it really works is it's a dose response relationship where bigger doses of virus lead to more risk of more severe disease. Same thing with aerosols. Like in a room, as persons exhaling them, since they stay in the air for hours, the concentration of aerosols eventually builds up over time. And eventually you get to some threshold where you meet that minimum level of infection. And then beyond that is just more virus. And you can imagine inhaling 200 viruses versus 200,000 viruses,
Yeah.
200,000 is a bigger risk for more severe disease.
Absolutely. And yeah, that's a huge thing. That's part of why, staying outdoors, keeping social distancing, all of those things are helping you potentially stay within some lower tier or threat of infection. So the actual infection mechanism, this is where I get nerdy in biochemistry, is these ACE2 receptors.
Yes.
And I don't even totally remember like the biological function of those ACE2 receptors, but this virus has keyed in to basically bind to this thing, inject its RNA. As soon as the RNA gets inside the cell, it starts co-opting proteins from the cell, forms kind of little or just like little invaginations in membranes where they're sequestering stuff like the ribosomes, and they start expressing that RNA. And as far as I know, I think it's like an open reading frame with like 16 different proteins or something like that. As soon as it starts expressing itself, it starts cleaving itself, which is an insane thing that, RNA can actually act as an enzyme, and like these proteins are coming out and processing it itself as it's still being expressed and making these viral proteins and stuff like that. So the biology of this is fascinating. It's just too bad that it's also potentially deadly and killing millions of people worldwide. But
yeah,
it's really
that's
cool.
a difficult thing is like, if you're a virologist or an epidemiologist, you are inherently academically excited by viruses. But the reason we study them is because they're dangerous. And so it's like, it's great, but we really need to stop it as soon as possible. But yeah, so ACE2 is actually, and this is a reason that COVID could be a little bit different from other viruses, is it's actually not just expressed in your respiratory tract. There is a gradient of expression of ACE2, that target where it's the highest in the upper airways, and then there's less ACE2 expression down in the lower airways. So we think the mechanism is you first get infected up here, the virus replicates until it's a pretty high viral concentration in your mucus, and then at some point you accidentally aspirate some into the deeper lung. And that's why you see a delay between initial infection and then spreading to more severe disease in the deep lungs. But that ACE2 molecule is also outside of the lungs. It's in your heart, it's in your gastrointestinal tract, it's in pretty almost ubiquitously throughout the body through reproductive organs. And so we think that could be a part of why we're seeing some of these consequences, potentially long-term consequences. Other than lung
organ
scarring,
damage, and I mean
heart
that's
damage.
the scariest thing about this is you're like, yeah, this might be like a bad cold, but like if you sustain permanent organ damage as a function of this, like it's a lot more than a bad cold.
Yeah, that's another good example of, you can't think about it binary as you survive or you die from COVID. There's a chance that you survive from COVID, but you have heart damage, which contributes to some percentage increase of heart failure. We don't know what that is because it hasn't been around long enough to tell. But you can't say, 99% survival means 99% totally okay. We just don't really have a good understanding of that yet.
Yeah, and that's the whole idea of like these comorbidities too, which aren't necessarily being counted as COVID deaths. But if you look at like the average death rate and then like the amount that were above the average death rate, you're like, okay, there's an extra 100,000, 150,000 comorbidities that are happening as a function of COVID as well because of those infection of other tissues. And I think that's a really interesting point too with the timeline of COVID infection is part of what has made this so insidious because it's cryptic. Unlike SARS or MERS or some of the other ones, which hit you and you get sick and potentially die within like a week. This is I have like a mild head cold for like a week and a half. And then suddenly, this turned bad and my deep lung is infected. And it's like,
right.
And it's not until you've been out in the world spreading it for a week or more that you realize that you're actually sick kind of thing.
Yeah.
And that's why we're having such an issue with this.
Exactly. It's like, how many days before having symptoms can you spread it? It could be several days, or you may never have symptoms at all. And that's on the other end of the spectrum from, right. If someone has Ebola or something, they have Ebola and they're not going to go to the grocery store, and the mortality rate may be much higher for Ebola. I don't know what it is, 50, 60, 70% or something, but it's harder to spread to more people because someone's bleeding out of their eyes. You're not going to go in the same room as them. With COVID, you might not be able to tell. And so it's sort of like, if you see a truck on the highway and it's got a big plume of black smoke coming out, all cars have exhaust. Most of the time you just can't see it. But if you're close to the one that you can see, you're like, I don't want to breathe that as much, but it's coming out of everyone. So it's kind of the same sort of balance with COVID's still here, but we just don't see it, so we don't worry about it as much.
Yeah, that's a good metaphor. I like that. So you mentioned Ebola, and that got me thinking, because I remember being in grad school when the Ebola outbreak was happening, 2014, 15, something like that. And we had a guy come in from Arizona who was part of like a rapid response team who had gotten out some Ebola vaccines with extreme quickness because they'd been working on them for a while. But what it was a cocktail of multiple things that it wasn't like one single thing that completely shut down the virus. It was things affecting the receptors, affecting the replication. It was like 3 or 4 things. And those doses are what saved some of the workers who'd gone to Africa and been infected, or someone, I think there was 1 or 2 people who came back here and potentially had infected, and they were safe. Completely done. And that was a triumph, absolute triumph of modern medicine. But in some ways, we'd had 20, 30 years to get ready for that because we've been working on Ebola vaccines for a while. And like you said, yeah, it was a virus that had a much higher morbidity, but is very hard to transmit. In a modern day society, you're not going to go near someone who's puking blood, and if they die because they puked out all their internal organs, you're probably not going to go touch and wash their body unless there's like cultural practices at play. So I guess I'm curious because there are some groups that have been working on corona type virus vaccines. Why is it taking us longer? And do you think it's potential that we're going to need multiple vaccines or a cocktail of things to try to fight this virus?
Yeah. Well, so part of our understanding from SARS and from MERS is why we've been able to roll out the vaccine so quickly now, is there were people who were actively working, particularly with the SARS vaccine. This one is basically just an updated version. SARS targeted ACE2 as well. This one targets ACE2. And so on the vaccine development front, they couldn't keep developing those vaccines because to test a vaccine, you need people being actively infected in the world, which luckily they actually stopped the epidemics in time. But what we can do is take some of the antibody products that were developed against SARS, and they've tested them, and a lot of them do bind against COVID, SARS-CoV-2. And so that has been the basis for the development of some of these, cocktails of different monoclonal antibodies. Some of them are derived from initially SARS patients. Some of them are derived from patients who have had COVID. You basically take their B cells, which are the cells that make the antibodies, sequence them, get the sequences of the antibodies, and then screen a bunch of different antibodies, find out which ones are best. And then the reason why you use a cocktail of 2 or 3 different ones is largely because you don't want, it's possible that one target of an antibody will mutate, and then that antibody won't stop the virus, and then you have the development of resistance. That's much, much less likely to happen if you're attacking 3 different components at the same time. Yeah. And so I think most of the antibody products that end up being developed probably will be a cocktail rather than a monoclonal, like a single monoclonal version.
Mm-hmm. And that's great. That's good to hear. That gives me some confidence that we'll be able to actually fight this, and that it's, that makes it less likely too that one mutation would suddenly make a vaccine less effective too. So that's exactly, that's encouraging to hear. Do you know if Moderna or any of the other ones that are kind of in those phase 3 things are cocktails, or are they all like single?
I believe those targets are single. So for the vaccine standpoint, I think it's usually a single spike protein sequence that they're putting in. Mm-hmm. I don't know that individual sequence though may well have separate epitopes. If you put in a big chunk of protein, antibodies can bind to different parts of it. And so you could develop, a polyclonal response against that single target.
Gotcha. Okay.
And so that's going to be the case probably for all, I think probably all of the vaccines in development. That's different from the antibodies, which obviously only have that single target. And so those will be a cocktail. But I think the vaccines should be inherently polyclonal because they give a big enough chunk of the virus that there's enough space for multiple different antibodies to bind.
Cool. That you just explained something that's very complicated in a very easy to understand way. So that your science communication.
It's the key. It's just a lot of random heads.
Let me see. What else did I have on here? So you told me you had a half an hour. Do you mind hanging out a little bit longer? Because there's still things I want to ask you.
Let's go a bit longer.
Cool. Yeah. So I asked you, a little bit of progress of vaccine timeline. So I guess what I see, and this is just my feeling without being on the inside of stuff, is that it seems like there's potential that by the end of 2020, we could have at least one viable vaccine. Worldwide, to get all the doses we're going to need more than one vaccine, because we're going to need vaccines of, like we just talked about, cocktails of things, different versions of things. We're going to need different companies producing different vaccines at different price points with different availabilities to try to get everybody vaccinated. And not that everybody has to be vaccinated, because there's a level at which certain people are sensitive or can't be vaccinated, or personal or religious exemptions or whatever. But we do need to get to a point where we have herd immunity, which is probably one of the most misunderstood things that's out there, and you've had some
Yeah,
great explanations. Do you want to explain herd immunity in your
sure.
patented way?
Yeah, so I didn't understand, and I was naive at the beginning of the pandemic. Herd immunity has a very defined definition in immunology, totally different from how it's typically discussed in the media, which I didn't anticipate coming. So herd immunity technically is just the threshold of people in a population who have to have immunity to prevent natural transmission of the virus through that population. And it's very easy to calculate. If each person who's infected typically infects 2 people, you need 50% herd immunity, because then instead of infecting 2 people, they infect 1 person. And so then the virus, it's not growing. It's just, a linear 1 to 1 to 1 to 1.
Right.
And so you achieve that through vaccination. If it was a 50% threshold or a 60% threshold, and the vaccine was 100% effective, you would have to vaccinate 50 or 60% of people. On the flip side, if the vaccine is only 60% effective, you'd have to vaccinate 100% of people. And we're thinking that the vaccine is not going to be close to 100%. And we're not going to get 100% uptake of the vaccine. So if both of those numbers are 60% and 60%, what we end up with is closer to like 30 or 40% immunity. And so one of the misunderstandings is that you have to hit herd immunity, and like that's the goal of the vaccine. But what really happens is it's a gradient of, especially if you vaccinate highly vulnerable people first, you're going to see a slower rate of infection through the population, and the people who are at greatest risk are now going to be at lower risk, either because they're not infected at all, or you can still get infected if you've been vaccinated, but you're at lower risk of severe disease. And so what's possible is that even if we only vaccinate 20% of the population, and it's the high-risk people, that's going to cut way, way, way down on the death rate. And so we don't necessarily have to get all the way to herd immunity. We wish we could. But like even a little bit is going to be super helpful by the time we get there.
That's well said. So I guess, so I mentioned, I think we'll potentially have a viable vaccine by the end of 2020, and that's rushing through all of the FDA guidelines and everything else. I mean, normally vaccine development is a 2, 3, 4 year process kind of thing. So we are extremely accelerated. We have all these phase 1, 2, phase 2, phase 3 trials happening in parallel rather than having them spaced out over time. And actually, to your point, there is a very, very good episode of Explained where they had it's a coronavirus 3-part special. And the middle episode is one that has a great visual representation of herd immunity and then all of these timelines that we're talking about with these vaccines. And I know you've talked a little bit to Cleo Abram on TikTok, and she's one of the producers for Vox, and I'm always blown away by their Explained series. They're so good.
Amazing series. Yep.
So anyhow, we might have some by the end of 2020. And what I know from my background in like looking at vaccine production is that scale up and production will probably say, well, we won't have something available for the public till maybe June or September of next year. Does that timeline make sense, or what do you think it's going to take to produce enough doses of the vaccine to get it out to the vulnerable groups and help us reach at least some basal level of safety?
Right. So it's going to be another circumstance where they've already got tens of millions of doses, or in some cases, hundreds of millions of doses ready, depending on which vaccine you're talking about. And so if one of those gets approved and we have, say, 100 million doses by the end of this year, then we're going to see if we get the data from the clinical trial in November, for example, or early December, then you're going to have to go through at least a couple of weeks, maybe a month of regulatory poring over the data to make sure it's safe and effective. But really, once that's over, those first 100 million doses are going to be rolled out to the highest risk people. And then they're going to continue making more and more doses. At that point, probably some manufacturing facilities that are currently being used for unproven vaccines are going to be shifted over into that like lead candidate. Mm-hmm. And so it's less of, we're going to wait until July and then vaccinate everyone. And it's going to be between January and July. There's going to be a fraction of people that just keeps increasing over time, starting with the highest risk. I don't have a good sense of the manufacturing rate, or, the fastest option would be all of the vaccines that are in testing end up getting approved, and then they've each already made 100 million doses or something. We could be good to go pretty quickly. Not on
and
a
it
global
depends
scale.
a lot on the mechanism of the vaccine, right? Those new RNA vaccines are phenomenal. And you could, in theory, if you can just produce enough nucleic acid, which is not a problem, we could have plenty of doses. Then it's going to be a function of like, how do you package it and store it and deliver it? And then it becomes more of a logistical question rather than a production question. But if we have to rely, if we have to rely on something like antibody production or something that actually requires a bioreactor to grow an organism and then a downstream process, that's going to take a lot longer. And I think there's potential too that there's, like I said, there's going to be multiple vaccines that hit that are at different price points. And we have, like, I think one of the things I'm really scared about is equitability when it comes to distribution of vaccine. Because, like, clearly Trump will get whatever's out there first. Right. He'll probably suck it out of a fetus or something like that. Whatever. He's Mr. Burns plus Biff, and he's every comical villain you could possibly come to life is our president right now. But
yeah, it's a good concern though, and I don't have a good understanding of, we say it's going to go to the high risk people first. I have zero understanding of how that's going to be managed on a practical level. Like, they'll ship vaccine to the states, which will be held in distribution centers. How do you tell who's high risk and who's not high risk? It's not like we have a database of people with hypertension or something like that. My understanding is that it's basically going to be either people's physicians recommending that they are high risk, but a lot of people don't have established primary care physicians. And so are people just going to show up and say, I have diabetes? I have no idea how that's going to work, but I'm interested in, maybe no one has an idea yet. I don't really know.
Or is it based on vocation? And does a company say, hey, we're going to give the state, a couple $100,000, and now all of our guys who work in meatpacking plants are going to get immunized so we can keep business going? And then it becomes, is it equitability, or
teachers or healthcare workers?
And
it kind of makes sense.
I think they should. I think teachers and healthcare workers would be better than guys working in meatpacking plants, But
right. Yeah, but you can make similar arguments. Yeah, it depends on who you ask. They could be like, well, people need to eat their meat or something. I really have no idea. I'm fascinated to see how it is going to play out. I don't know that there is like a publicly released game plan of how that's going to go. They did, the HHS did say, I think 2 months ago, that they told the states basically to be ready for a vaccine as early as November 1st. And a lot of people got very upset about that because they assumed that they were rushing it before the election. But what they were really saying was figure out your distribution centers, because we don't have a national network capable of, managing the supply chain to vaccinate the entire country.
We've never done something like this before. We're used to sending people ballots. We know how to send a census. We've never done this with vaccines or something that, of the scale and the measure that's going on. So.
Right. So what we really don't want to happen is to have the vaccine ready, to have 600 million doses, but not have the network needed to distribute it. That would be quite sad.
Yeah, the healthcare workers and the people administrating it, and there's a lot that goes into that.
It's a logistical, incredibly complex, but people are very good at their job in supply chain. So I assume it's going to work out, but I have no personal understanding of how that is even managed.
So I guess what I'm thinking about is certainly looking towards institutions like the NIH and Francis Collins and Dr. Fauci, Dr. Bright. There's been some scientists who have come out as absolute heroes and role models and inspirations, I think, to normal Americans who still believe in the experts. And I'm just curious about if you have any thoughts on like institutions like the NIH and what role they have to play in getting normal Americans to accept a vaccine and to want to try to move beyond this disease. And ideally, like, we have to depoliticize this entire thing, because there is no greater good than public health. And right now it's been politicized and we're literally killing people because of the politics. Like, that's got to stop.
Right. It really, which is fascinating, because this hasn't really been an issue up until now, really. And so to a large extent, I don't think the NIH or the CDC or the World Health Organization were really prepared on the communications front. They all have communications agencies. They maintain an Instagram. They put out their infographics. But it's way misbalanced toward misinformed people posting things on social media versus actual credible organizations. It's got to be like 10% or like 5% of the volume compared to the misinformation.
Yeah, totally.
And so over time, that just wedges this divide between people who hear politicians say something which matches the 95% of incorrect stuff they see on social media, and then the 5% that they maybe they don't even see it on social media.
Yeah, they don't even get exposed.
Right. If you have to seek it out. You live in this bubble in your social media world with whether it's the TikTok algorithm or the Facebook algorithm, whatever it is, the news channel you watch. And so it's very difficult. I think we didn't anticipate the need for both the development of the science, making the vaccine, or the therapeutics, as well as communicating to people. It's equally important, because it doesn't matter if you have the vaccine. If people don't get it, what good does that do you?
And if they don't trust it, if they think that it's implanting a microchip under their skin so the government can track them, which, by the way, is your smartphone already.
Right.
But I guess that's where I heard a call several months ago about something about like, hey, if you're a scientist, now is the time to amplify the message of not just truth, but just like, here's what science is. And I think in some ways it's really difficult because misinformation is so much more exciting. It sounds so much more, you're like, oh, I have this hidden secret that doctors don't know about. And you're like, yeah, no, doctors know about that. That's not a thing, but it's
right. But you're going to hit that share button.
Right. Trying to debunk stuff and trying to tell the truth is so much harder than just sharing misinformation. And it's always the idea battle. In any argument, it's easier to push away and say, no, that's crazy than it is to actually hear the argument, synthesize it, and try to understand it in your own context. And certainly for people who don't have like a science background, the difference between like an RNA versus a DNA virus is meaningless. To me, right. Because I have a background, I think it's fascinating. I got interested in the biology of it, but it's helped me understand everything along the way and make my own inferences. But I have a PhD in this stuff, and not everybody does. So how do you reach people? And I guess that's where I'm like, your account is something that I think people should be holding up, be amplifying. I'd love to see like more of you on the news. Like, can we get you as a talking head on CNN or whatever it is? Like, we need people
I would be
like
open
you
to doing it.
Yeah,
it's an uphill battle that I've faced that, I know 100%, I could just make something up totally wrong about coronavirus, say it very convincingly and get 20 times the exposure I get when I make like a regular video about the true things about the coronavirus, just because people are like, oh, that's scandalous. I'm going to share it. See what people think about this. That's always you see that reflected in the fact that people who have no experience at all, very clearly, either because they're 14 years old, and which, you're not going to have TikTok. PhD level experience to make claims about the mortality rate of coronavirus at that stage. But, people do it and it goes incredibly viral. And it's really hard. It feels like you're always 3 steps behind, because then you have to identify that misinformation and then you try to make a counter video and it gets like 10% of the views. So it's really hard to be sort of in front of that. Yeah, partially because you can't predict exactly what people are going to make up ahead of time. Like the whole 6% thing with the CDC data. If I had, just read that data, I never would have been like, people are going to interpret this as though 94% of the deaths aren't due to COVID. It wouldn't have even crossed my mind until it's out there. And then you're trying to catch up to this thing that's so obviously not true, but like everyone's heard it. And now people are kind of questioning it, even if they don't fully believe it.
Yeah, that's really hard. I've been thinking a lot about recently of like, how can we make like real conspiracy theories? How do we turn real information into conspiracy? Like, and I had this thought over like global warming. Like, oh, what if global warming is aliens that are like shooting a ray at our polar ice caps and melting them because they're trying to cause a mass extinction? Could we then unite against global climate collapse if we thought aliens were doing it? And I was like, I don't know.
I think it's a good idea. Yeah, anything that works, honestly. It's a good idea. Like, try to capture some of that intrigue and like scandal, but have it be actual information. And a couple of people tried that I saw on TikTok with, like masks. I forget exactly what they said. It's like the government is trying to get you to not wear masks so they can read your lips with CCTV cameras or something. So wear your mask so they can't invade your privacy or something like that. Hey, if it works and it prevents someone from spreading it to someone's grandparent, whatever.
Yeah. I mean, to me, the amazing thing is still that like the recommendations have pretty much been the same since the start of this thing. The only thing that shifted a little bit was in the first like month, they were not sure about the mask thing because we didn't know as much about transmission. And honestly, at the time, there was a real, people were stockpiling TP and everything, and they were afraid that people were going to stockpile masks and there wouldn't be enough for healthcare workers. So they were like, maybe you don't wear masks. And then very quickly afterwards, they're like, yeah, no, you should definitely wear a mask. And so wash your hands, wear a mask has continued to be the advice for this entire thing. And yet there's still so much pushback because I think of cultural and political factors. And it just, again, public health helps everybody. You lose nothing by just wearing a mask. And I think there's a lot of healthcare workers out there who get really frustrated by people who come up with these ridiculous arguments of like spraying a hose through a mask and saying, look, it doesn't work. Like, it's not.
Yep, that's not a rigorous study. Yeah, you raise a good point though, that a lot of the actual actionable advice hasn't really changed over the pandemic. And so that's one say when people ask me about like, how do you stay current with the news about COVID, or like I'm stressed and anxious about this. A lot of the time my answer is you don't really have to follow, like it could be a full-time job to stay current with coronavirus, but you don't have to. All you have to know is about when there's new actionable information. And the most recent actionable information is that ventilation is probably important when you're indoors because of the potential transmission as aerosols. But other than that, wearing a mask, washing your hands, staying apart from other people, usually being outside, having good ventilation indoors, if you're doing that, you don't have to read anything else pretty much about coronavirus, because it's not going to change what you're doing. And so it can be very mentally draining to be thinking about the pandemic all the time. But as long as you are taking the necessary precautions, you don't have to stay current. There are people who are, believe me, there's plenty of people who are staying current with it so that when there is a new actionable thing, you'll know about the new actionable thing to do. But it's easy to get wrapped up in the 24-hour news cycle of this mutation is making COVID 10x more infectious or something, which is often misinterpreted anyway. So
yeah. Well, let's take a deep breath, because you had some mushroom questions for me, and so I want to jump into that, because I think you had some thoughts that sounded kind of cool.
Yeah.
And I know that you'd expressed to me that you'd never really seen mushrooms in the way that you'd seen them like on my page. So do you have any history with mushrooms, or like what got you interested?
No. So my history with mushrooms is nothing. Is buying them at the grocery store. It's no academic background. Not even much. Like, I didn't know that there was mushroom entertainment content in existence until really I came across your channel on TikTok. And so it's kind of cool to see, like, I didn't know that people like went mushroom hunting, for example, but it's an awesome hobby. And so I've got one question is, the ones that you see, what is the relationship between the mushrooms that you see and the rest of the fungus, basically? Like, is it a small fraction? What fraction of it is actually above the ground? What fraction of it is below the ground? Is it like an iceberg effect?
That's a great question. That's a great sort of thought about what constitutes fungi. And I guess I can start by saying that the mushroom is basically it's what's called a fruiting body, but it's akin to like a flower or an apple. It is the reproductive organ of the actual sort of mushroom, which is the mycelium that lives below. So the mycelium is, there's a great book, then I can send you this recommendation later, but it's a guy, Merlin Sheldrake, who I talked to a couple of weeks ago, and he wrote a book called Entangled Life, where he went into all these different factors about mycelium. And he has a quote there, something like, mycelium is a way of life that challenges our animal imaginations, because mycelium is this growing in all directions. It's a complex network that is, there's no nervous system, and yet it's able to make complex decisions across this network and reform itself and go after particular substrates and make all of these insane, complex intelligence decisions with no centralized intelligence or rhyme or reason to it. And so mycelium is spread out through whether it's soil or dead wood, and that depends on whether we're talking about something that's a saprobe, that's like degrading dead stuff, versus something that is a mycorrhizal, so it's in association with a plant, or something that's parasitic. So there's different types of having mycorrhizae, but ultimately the mushroom, the thing that you see, is when the mycelium conditions are right, and it decides, hey, we're going to like put a bunch of our resources into reproducing. And so we're going to generate this thing to spread spores. And there's enormous biodiversity in the way that fungi are able to spread those spores. And that's what you see as the diversity of mushrooms, which is incredible, because your sort of standard mushroom is like a stem and a cap, and spores come off of that. But even in that, gilled mushrooms have been evolved multiple times through lots of incidences of convergent evolution. There's been things that lost their gills and then re-evolved gills. There's things, I'm sure it's somewhere you've seen time lapse of like those crazy stinkhorn, like a red octopus thing opening up and covering all this like black nasty stuff. And that's like, that's this gleba that smells like rotting flesh, and it's attracting insects to help spread the spores. And so that's another sort of, it's not quite a zombie. That's cordyceps.
Okay.
It's just amazing how like mushrooms have co-opted the natural world, just like a flower has, to promote animals to come and help them spread their spores and, disperse their genetic information into the world. So that's pretty cool. But
I see a good question in the comment. How many types of edible mushrooms are there?
Oh, Lord. There's something like maybe 40,000 different kinds of mushrooms that are out there. And I'm talking about fungi that produce mushrooms, because there's a whole other set of fungi, and I'll talk about that right after. But sure. Within that 40,000, there's probably like 200 or 300 pretty good edible species on one end, and maybe like 200 or 300 like pretty poisonous species on the other end. And everything in between is not edible, not poisonous, not psychedelic. It's just a mushroom. And that's, humans love to try to categorize things, and fungi just like do not care about categorization. They don't want to fit into the nice little things that we try to put them into. And so
So it's not
that's
edible then just because it's not delicious, or if it's not poisonous, I mean, you could eat it, right?
well, I mean, a
It's
lot of
just
them
you
are
don't
like,
want to.
there's like woody polypores. Could you eat a piece of wood? I mean, you could try, but you're not going to
No,
get very far.
not typically.
So yeah, I mean that's part of it. Is yeah, they're very very small. They could be really like gooey. They could be there's a lot of things that are just inedible, and it's not because people love that adage of like, well, every mushroom's edible once, which is I think it's a really stupid sentiment because yeah, you could eat something and die. That's fine, but like not everything is edible. You would have a very very tough time eating a lot of mushrooms. Just I mean, and to the point, this poster behind me, these are all edible mushrooms. These are all toxic mushrooms. So that, there's lots of each kinds, and this is hardly an exhaustive list, but just there's a lot of diversity in the types of things that you can find. And certainly within like the fungi kingdom, there is a real divide between what counts as like a mushroom versus what's just a fungi. So there's a huge number of molds, of yeasts, of small fungi that exist in the environment. Fungi are ubiquitous. They are absolutely everywhere. They're all over your apartment. We could probably put out a little glass of juice somewhere in your apartment and see fermentation within a couple of days. And that's not because your apartment's dirty. It's just because fungi are freaking everywhere, and their molds
Right.
are everywhere. You leave a piece of fruit out, you leave bread out, you're going to get mold. They're just everywhere. Usually they're dependent on water activities. That's the biggest thing you can do to inhibit fungal growth is just make it too dry or too hot, too cold, that kind of thing, which is pretty standard for microbial growth. But within that, so there's this huge diversity of molds and yeast and other things. And then within mushrooms, we have like mycorrhizal mushrooms, and there is a split between what's called ectomycorrhizal, which is things that are associated with plants and producing like large fruiting bodies, versus stuff that's like arbuscular mycorrhizae, and those are very small microscopic fungi that live like sort of in between plant cells and still form mycelium and mycorrhizae, roots into the soil, but they're not producing sort of large fruiting bodies. And so there's like 85 to 90% of plants on Earth have a mycorrhizal partner, but not all of them are ectomycorrhizal fungi that produce big fruiting mushrooms.
Okay. Do the other 10% or 15% of plants share some sort of common, like, is that a certain type of plants that don't have that relationship, or is it just kind of at random?
They are legumes. So they
Oh,
are
really?
forming little nitrogen nodules. So they're inviting bacteria in to basically invade this nodule, and then the plant is changing basically the redox conditions of that nodule to trick the bacteria into being its own sort of symbiont to fix atmospheric nitrogen for. And that's, my dad has worked on that at MIT for years, so that's part of the reason I know that whole narrative. And actually, interestingly, the heme protein that those bacteria use in legumes is what they've expressed in yeast to make like the Impossible Burger red. So that's like science is just round and round. It's cool.
Okay, so nitrification, denitrification is crucial in wastewater treatment plants. Usually that's accomplished with bacteria. Are fungi used as part of that wastewater process?
I don't know. I don't know that much about like wastewater processing. I do know, I know that they use redox potential, which is one of the things I do my research on, as a process parameter for when something has been sterilized or not sterilized, because it's dictating what types of microbes are able to grow and how they run their metabolism.
Right.
But certainly with the idea of like fungal bioremediation, which is something that's really sexy and really interesting, but it's not fully fleshed out about how that actually works. Like we can show in a lab, you can get like oyster mycelium to eat hydrocarbons or eat like a cigarette butt. But if you take that oyster mycelium and dump it on an oil spill, it doesn't clean it up because it's like, oh, there's dirt and organic matter here, which
That's much better.
way more interested in eating than oil, which
Plastic.
causes, yeah, exactly. It's a very different metabolic cascade of stuff that leads to that. So there may be some need to do genetic engineering or other ways of like selecting and tricking fungi into doing what we want them to do. But we have a long history of that. I think people don't realize that like a lot of the commercial enzymes we produce for stuff like laundry detergent is coming from fungi. And we've been doing this for 40, 50 years. And we just, there's so much more in the fungi kingdom that we can kind of like ally with. And I think people really need to recognize the importance of it in agriculture and farming, in the environment. The ectomycorrhizal mushrooms I mentioned, they're generally associated with like large temperate forests. and they're responsible for sinking something like 30 or 40% of like the carbon on the planet potentially, because their mycelium
Wow.
is so good at sinking carbon. Like 50 to 70% of the carbon in a forest is actually down to the mycelium kind of thing.
Oh, wow. I didn't realize
So
that.
it's, yeah, I mean, again, these stats are a little, I could like, the studies are
But they're
fairly
also
recent and we
codependent
never know,
on the
but
trees, right? Or
they
could they
are,
exist?
because
So you
no, because
can't just get rid of
it's
the
a
trees and
system.
then
No,
just have
you can't
the.
get rid of the trees, you can't get rid of the fungi. And I think one of our issues with like agriculture and growing things in general is that we've forgotten how important the fungus was, because there is this constant exchange of, the trees producing sugars, sending it down to the fungi are absorbing minerals and nutrients and sending it back up. And there's this kind of give and go. And it's not always, it's very much like a push and pull kind of thing, because these relationships have been formed somewhat antagonistically throughout all of evolution, and they're very much codependent, but there is a push and pull. And even within like the season, when a tree has leaves, it's producing a lot of sugar, so it's sending those sugars down. When the tree doesn't have leaves, it might be pulling up from the fungus. So there's like these really interesting relationships that go on. And then within that, it's not just one tree and one mushroom, it's many different species of fungi and
The
mycelium in the
network.
soil, the network of the entire forest. So all of these trees are exchanging carbon, and mushrooms are kind of like the translators between the trees, as they're like, dictating this knowledge and exchange of nutrients and knowledge, as it were, across this entire network. And so, again, we as humans have reduced everything down, like I was talking about, the siloing of different departments. And now we're starting to realize, like, oh crap, nature really is all connected. And
It's far more complex than we thought.
Exactly.
We can't
just
reduce it to
isolate
one
things.
textbook per field.
Bingo. So, yeah, it's pretty cool stuff. And I think you mentioned, one of the things you asked me about was spore dispersal. And so I talked about like stinkhorns and having little nasty gleba stuff. And there is some recent evidence that mushrooms are almost creating their own convection air currents through like the evaporation of water, basically. But beyond, you can, and there's some nice like photos and things of a mushroom sitting there with backlit. You can see the spores kind of cycling off it in this little way. But there's other fungi that literally shoot their spores out. So there's these little things called cannonball fungi, which are a nuisance for homeowners because they grow in wood chip beds. And they're just these like tiny little orange dots. But they, through the action of like turgor pressure and other things that are going on with the membranes, they're able to kind of explode and literally shoot their spores like, up to, I think, like 10 or 15 feet kind of thing, which for the fact that the organism is like that small, that would be like a human being like throwing something to another state.
Several football.
Yeah. So it's that kind of thing is just really cool. There's so many examples of interesting things like that in the fungi kingdom. It blows my mind. And the other thing too is mycology is very under researched. There's a lot of stuff still to be discovered. Big pharma hasn't necessarily bought into all of the bioactive compounds, the polysaccharides, and the things that are there. And I think it's hard because it's natural products and so on. But I am hopeful that we'll see people exploring more compounds from mushrooms for medicine. So
Right.
maybe someday you'll work on a mushroom.
Yeah, maybe that's more the chemist. That's more the chemist side, which I can appreciate, but from a distance, because the actual screening and isolation of those compounds is a skill set beyond mine.
Cool. Well, it's been an absolute pleasure, and I'm really happy you took the time to talk to me.
Yeah,
you,
no, this
Doctor.
was great. Enjoyed it a bunch. What I've learned is I've got a lot of reading about mushrooms to do because this is some cool science.
Yeah, I'll send you. I can send you some links or a couple of book recommendations, something like that, if you want kind of a starting point. But
Please
honestly,
do.
I'd
That'd
say
be great.
just go on social media, go on Instagram. There's some great accounts out there. There's people sharing all sorts of good science, and I think in the same way that we have a need and a desire to share curated scientific information about whether it be COVID or mushrooms or whatever, this is a call on anyone who's a scientist and has the capability to look at a camera and talk. Get out there, tell your story, tell people what you know about, because it's never been more important to like spread good information and try to fight that misinformation. And ideally, we can make it even more entertaining than the misinformation to some degree, so we get people to listen.
Yeah, there are far too few of us. So if anyone watching has been thinking about doing it, just do it. Start posting, whether it's just for your friends and family or for a wider audience. Give it a shot.
Awesome.
Okay,
Thank
Gordon,
you so much,
great talking
Doctor.
to you.
Yeah, you
Talk
too.
to you later.
Have a good one. Adios.